So what if I told you that for a whole lot of people, hearing loss has nothing to do with loud noises or getting older? What if it's all down to a single tiny typo in their genetic code? Today, we are going to do exactly that. Decode the blueprint of our hearing, see how it's written, and find out what happens when there's a mistake.
It's a pretty wild idea, isn't it? The thought that something so huge, so fundamental to how we experience the world, could boil down to a tiny, inherited glitch in our body's instruction manual. And that is exactly what we're going to unpack together.
All right, here's our game plan. First up, we'll get a handle on our basic genetic blueprint. Then, we'll see how these tiny changes in the code can get passed down. We're going to look at cases where that typo only affects hearing. And then we'll look at others where the impact is way, way broader. And you'll want to stick around for the end because we're going to look at the mind-blowing science that might one day let us literally rewrite that code. Let's do this.
Okay, so to get a grip on genetic hearing loss, we've got to start with the basics of genetics. And honestly, the best way to think about it is like a giant, super detailed cookbook for building a person. So, inside pretty much every single cell in your body, you have these things called chromosomes. We've got 46 of them. Think of these as the chapters in our cookbook. They're the big organized binders that hold all of our genetic information together. Now, if the chromosome is the chapter, then a gene is a single recipe inside that chapter. Each gene has the exact instructions for building one specific thing our body needs. Usually, that's a protein that has a very particular job to do.
And just how many recipes are we talking about here? Well, scientists figure we have somewhere around 20,000 different genes. That's 20,000 individual instructions for building and running you. That is a lot of recipes to get right every single time.
So what happens when there is a typo in one of those recipes? And how in the world do these changes get passed on from one generation to the next? Well, that brings us to the really interesting stuff: inheritance and mutations.
So think about how we inherit things. For what's called a dominant trait, you only need one copy of that gene from one parent. It's basically a 50-50 shot. But for a recessive trait, and this is key for a lot of genetic hearing loss, you've got to inherit that specific gene from both parents. And that's exactly why these traits can seem to vanish for a generation, only to pop up again later down the line. Now, these typos aren't all the same. Some are really small, like a single mistake in one recipe. Others are huge, like having a whole extra chapter or missing one. And while those massive changes have, well, massive effects, for our story today, we're really going to zoom in on the impact of those little single gene mistakes. This is where it gets really specific. We're talking about a typo that affects hearing, and pretty much nothing else. It's called non-syndromic hearing loss. And believe it or not, it's the most common form of hereditary deafness out there. And get this, it's not a simple one-and-done story. Scientists have found over 100 different genes where a single mutation can lead to hearing loss. It's this incredibly complex network of genetic instructions all working together just so we can hear the world. But even with those hundreds of genes, one of them is by far the main culprit: a gene that makes a protein called connexin 26. Mutations in this one gene are responsible for about half of all these cases. That is just staggering. One gene accounts for 50% of the problem.
And this just shows you how incredibly precise this science has become. It's like genetic detective work. Researchers have been able to pinpoint exactly where this critical gene lives, right there, on a specific little stretch of chromosome 13. They can go right to the scene of the crime.
So why is this one little protein so important? Well, connexins are basically the construction workers that build tiny tunnels between the cells in our inner ear. And these tunnels are absolutely essential. They help recycle potassium ions, and that recycling process is what allows the delicate hair cells inside our ear to turn sound vibrations into electrical signals our brain can actually understand. So no tunnels, no signal, no sound.
But what happens when the genetic typo doesn't just stop at hearing? Sometimes a single faulty gene can set off a chain reaction, causing a whole bunch of issues all over the body. And this is what's known as syndromic hearing loss.
And the variety here is just immense. We're talking about more than 300 known syndromes that list deafness as a symptom, often right alongside issues with the heart, the kidneys, the eyes, you name it.
Let's look at a classic example: Pendred syndrome. It's a recessive condition. Hearing loss starts really early in life, and it's often linked to a specific physical change in the inner ear. But here's the twist. It can also cause problems with the thyroid gland, which might not even show up until someone is a teenager or an adult.
And yep, just like before, scientists have hunted down the cause. It's mutations in a different gene, this one called SLC26A4, which lives on chromosome 7. It's the one link that connects problems in the ear and the thyroid all the way back to a single genetic source. Okay, so knowing the cause is one thing, but can we actually fix it? This brings us to the most exciting part of this whole story, the future. We're talking about the incredible potential of gene therapy.
At its heart, the idea is just so powerful. Instead of just treating the symptoms with drugs or with devices, gene therapy aims to go right to the source and correct the underlying genetic mistake itself.
So how does it work? Well, one of the coolest methods uses a harmless virus, think something like the common cold, as a delivery truck. Scientists basically hollow it out, they take out the viral genes, and then they load it up with a new, correct copy of that faulty human gene. Then they use that virus to deliver the correct recipe right into the target cells. If it works, the new gene just starts doing its job, making the right